important. Oceans absorb up to 50% of the anthropogenic CO2 released. Okay, so oceans is now a major sink of the CO2 that's released by human activities. That's why we need to understand the processes that occur in the oceans. Only through understanding that we can hopefully uh, increase the amount of CO2 that can be absorbed, or so that we can uh, reduce the impact of increasing CO2 in the atmosphere. Okay, so later on we'll talk about the physical and biological processes that actually drives the CO2 transfer into the sea. Okay, just imagine if the oceans are absorbing up to half of what we are releasing, half of the CO2 that we are releasing. Okay, and if you remember the Great Ocean Conveyor Belt, it takes up to 1,000 years to complete one loop. So sooner or later, all the CO2 that's being absorbed will be released back into the atmosphere. That's why uh, the climate scientists are always worried that we do not understand the tipping point, which means when the change occur, it is not going to change incrementally. It is going to be drastic. That is why climate scientists are worried. Okay, we are worried that the conveyor belt might stop. We are worried that all the CO2, all the pollutants that are being trapped in the deep waters will be released after once it is near the end of the conveyor belt cycle. So a lot of things that are uncertain. That's why understanding the oceans will help us better predict what will happen with the continuing increase in CO2 emissions. Okay, so that's how you relate the the importance of ocean in relation to the climate change problem that we have. So we, of course, when we talk about marine and estuarine microbiology, it is, of course, we're talking about a field science. Okay, so when we want to study the environment, as I mentioned earlier, taking the sample is important. So we have essentially covered the grid sampling, the linear transect, the random sampling in microbiology. So of interest here in this course is we are also tackling estuarine and also riverine waters. So just a quick question here. If you are asked to sample a river, do you sample going upstream or going downstream? So, if you remember, uh, river water also flow downstream. The problem is if you sample from upstream going to the river mouth, you may be sampling the same water. Do you get what I mean? Because maybe if the river flows slowly, then by the time you reach the second station, the water that you sample upstream would have moved to the station that you're sampling again. So you will be sampling the same water. But that there is a danger in that. So usually the best practice is actually to sample downstream going upstream so that you do not uh, get the samples mixed up. Okay, so that is the reason, uh, that is the main reason for why we sample from the river mouth going upstream. Next, within the context of oceans, okay, we Malaysia, we have very shallow waters. So we do not have much of a problem when we want to sample the the sea. Okay, we don't even have an ocean. We only have seas. So usually we take the surface water, then maybe take 10 meters interval. That will be enough because most of the uh, water depth in the sea around our peninsula Malaysia and is only around maybe 20 to 50 meters deep. So it's not very deep. So there is not much problem with seawater sampling. Okay. However, when we talk about oceans, then you've seen from the geomorphology lecture, you've seen from the figures that most of the oceans can be up to 4,000 to 6,000 meter deep, 2,000 to 6,000. So how do you sample? If you want to know a water column profile, let's just say you are asked to study a water column profile. A water column profile means you want to study from the surface onto the bottom of the sea, but at that station. So how are you gonna sample it? Are you gonna sample every 10 meters? That is impossible, right? So there has to be a kind of preliminary assessment or preliminary data that to help you decide how to at what depth you should sample in Peninsular Malaysia shelf, continental shelf, the Sunda shelf. So we have the Sunda shelf. So that no problem, maybe about 50-meter depth. Okay. So easily you can say you do every 10, 20, 30, 40. You sample every 10 meters. That will give you a very good water column profile. So this is uh, okay. So sometimes when you look at oceanography journals, you see scientists that are doing profiles like this. Okay? But when you look at the oceans and you are tackling up to 2,000 meter deep, okay, there is no way you can do every 10 meters. So you have to use your general knowledge about the oceans to come up with a suitable sampling plan. Okay, the first knowledge that the first knowledge we have is we know that there is an euphotic zone. Okay, euphotic zone is where there is enough light for photosynthesis. You know that photosynthesis, the primary producers are the main component, the main producer in the food web. So if therefore the euphoric zone is should be very important. Okay, so how do you measure where is the euphoric zone? Usually you can measure this either through light. Okay, light transmission. Okay, so you should measure with a lux meter to see how not with a lux meter, with a PAR. They use a PAR. It's a photosynthetically active radiation. So it measures the photosynthetically active wavelength of the light. Okay? then you will know where is the euphoric zone, where is the dysphotic zone, and where is the aphotic zone. Also, you what how scientists will usually start off with a TS, temperature salinity profile. Okay? Temperature salinity profile. Okay? So this is easy. This is just by placing the probe going all the way down. Here you will see that the waters are usually this. Warm waters near the sun upstream, and then go down and all the way. Temperature profile, you can guess that the euphoric zone is here, where it's warm enough for most of the biological activity. So you can focus more on the upper waters, maybe 0 meter depth, then 10 meter depth. Then you can actually go down to 50 meter depth, and then 100 meter. And then afterwards, you can just go to maybe 500 meter, 1000 meter, then 2000 meter. So when you have a deep water column profile, focus is always on the upper waters where most of the biological processes occur. Okay? Because once the waters are very cold and below the euphoric zone, biological processes are slower. There shouldn't be much changes in the uh, chemistry profile, in the biological profile. Okay? That's why we can space it further as we go deeper into the water column. So for oceans, this is how you should plan your sampling. Whereas for rivers, it is from downstream, from the river mouth going upstream. Okay. Any questions? Any questions? Okay, so okay. So when you look at this, can you imagine the amount of effort to sample up to about around 2000 meter deep? Okay. Sampling means you have to put down the sampler. Okay. Slowly let the sampler go down and then bring it up again. 2000 meters, that's 2 kilometers. Okay. So every sampling will take a long time. Okay. You cannot be, because even the movement of the sampler down the waters will take time. So sometimes up to 2 hours or more, depending on how deep it is. So if one station takes around 2 hours or more, that's why when oceanography work, a lot of planning has to take place because you cannot be wasting your time because ship time is expensive. Okay, very expensive. The ship itself is expensive, so different countries have different uh, policies. So in Japan, uh, the universities will have one big ship, one big cruise ship, then maybe a few small ships for those who have programs in oceanography. In Taiwan, the universities don't own the ships. In Taiwan, only the government owns the ship, which means the government builds the ship and then the ship time is shared among the universities. So every year there is a bidding for ship time. Okay, so there will be a panel of evaluators. They'll look at the quality of the proposal, what you plan to do, and then they'll decide whether to give you ship time or not. Okay, so it's competitive to be able to go onto the uh, ship. So in Malaysia, we only have one university with a research vessel. That's the University of Malaysia Terengganu. Okay, they have fisheries course there, so they actually use the ship to train uh, fishermen, um, more like offshore fish, commercial fishermen. Okay, the ones that go to the Indian Oceans and Pacific Oceans. So they need to know how to handle ship, uh, the equipments on the ship and all that. In Sabah, yes, in Malaysia, Sabah, they have a more of a yacht, a 12-person yacht. The maximum is 1 week cruising, which means you can go offshore for 1 week and then come back. Okay. Not that far because the ship is more of a yacht. Okay. Okay. In we don't have our own ship, although we did rent a research vessel at one time in 2004 to traverse the Straits of Malacca up and down. Yeah. So okay. So if you so there's a bidding for the ship time, and then you'll usually have physical oceanography, chemical oceanography, and biological oceanography. Okay. So all three components will usually be studied together. So imagine you also have to share the water samples with the chemical oceanographer. The physical oceanographer usually just works with sensors and measurements, on board in situ measurements. Okay. The ship time is expensive because you have to pay for the captain, have to pay for the sailors. All these incur charges. That's why in Malaysia only one university has the research vessel, because you we not many universities can find funding to maintain a ship. Okay. Okay, this is a Rendon design. Can you see the slide?
No,
No. Oh, you can't see the slide. Okay.
I have to change it.
Pardon? Can you see the slide
Yeah,
now?
yeah.
Okay. So this is the Rendon design. This is the simplest water sampler. Just two suction cups here, a trigger here. Okay. So what does the trigger mean? The trigger is just that when you see the wire here. So when you place a weight here, the weight will hit a button, and the button will release these two suction cups. So these two suction cups are pulled inside with an elastic band. So once this is released, the two suction cups will clamp onto each other. Okay, so that will trap the water that's inside. So what we usually do is we open it up like this, we set it up like this, then we lower it into the water, and then we drop the weight onto this area here, and that will trigger the release of these two suction cups that will close and capture the water inside here. Okay, so this is the simplest design. Okay, this is the Vendon design. So because it's horizontal, it's ideal for to sample just above the bottom sediments. It's ideal for when you want to study layers of water, okay, different layers of water. However, if you look at this design, there is no weight that you can use to there's no way you can weigh down the sampler. That means this doesn't work well in the oceans, this doesn't work well in rivers where there's river flow, where there's currents. It will sway around, it won't stay in one place. So it's not suitable for waters with movement. It's more for uh, lakes, or slow moving streams, okay, or stratified water bodies. This is the more conventional water sampler. This is the Niskin water sampler. Okay. Same uh, principle with the Vendon, but more robust. Okay, so the weight is dropped also here along the wire. Once it hits the trigger, it will close and you will trap the water inside. So when you are required to take many samples or at many depths, it is not feasible to use only one Niskin water sampler. So what they usually do is they will have a multi rosette mounting bracket where you can put different a few samplers onto the mounting bracket so you have a multi water bottle sampling system. So this is triggered electronically, which means you don't have to place a messenger, a weight here. So once this bracket has reached its required depth, they can trigger it electronically, then one will close. Then the next step, the other will close, and so on and so forth. Okay. So this is for a larger form of sampling. Okay. This is a Lymnos water sampler. Okay, so you see this one, you still have to open it up before you put it into the water. This one you have to open it up before you put it in the water. Okay, but this one you don't. Okay, so this is a Lymnos water sampler. This is simpler. So it is lowered closed to prevent contamination by other water layers. So only when it's reached the desired depth, you can trigger it so that it opens up and water can go in. Okay, so this is the definition of a COC, close, open, close sampler. Okay, close, open, close sampler. And your Niskin are open, closed samplers. Then your GoFlow and the Lymnos are your close, open, close samplers. Okay, So you close it first before you put it into the sea, and then when you reach the desired depth, you open it up. Okay. Why do we require it to be like this? So you open it up only when it is in the sea. Then once you reach the required depth, you trigger it to close. But why? Because the surface water is the most polluted. Okay. The surface water here, the surface layer here is where there is most pollution. The oil sheen, okay, there will be layers of pollutants, the heavy metals, all the particulates, all the particles tend to uh, accumulate at the surface layer. Therefore, you have to, for oceanography, especially those that are studying trace metal. So when you're studying trace metal, you are actually talking about parts per billion concentration. Trace metal, for example, they will be studying vanadium, they will be studying cobalt, cobalt concentrations, uh, zinc. Okay, so these are all considered nutrients. So those for the phytoplankton. So there are a group of scientists, geochemists, who are interested in trace metal studies. So they also iron, yes, iron also. So you want to measure trace metals at parts per billion level, you cannot have contamination like this. Okay? So if you were to drop your Niskin sampler through this surface layer, most of the contaminants will be stuck onto the sampling vessel. So it is there is no way you can do trace metal studies if you were to use this approach. It has to be a go flow closed metal closed open closed sampler. And also for trace metal studies, usually they require a clean room. A clean room is something like the biohazard room where you have to wear special suits, and then also remove your you have to wear coverings for your shoes also, because and then you have to enter a small chamber where the air will flush all the particulates or any particles that are attached onto your yourself. Okay, you have to wear also a hair covering because you do not want your dandruff or anything to contaminate your samples. So that's how clean you need to be when working doing trace metal studies. Okay, so yeah, because even the dust on your shoe, the dust on your shirt and everything can uh, affect the results that you get from this. Okay, that's why you have to use very clean methods to seawater. For example, you can differentiate it into particulate or dissolved. So this is a kind of an arbitrary definition. So because in nature there is no real cutoff point between particulate and dissolved. But for scientists, we set it depending on the study that you want. For example, if you're looking at organic carbons, some scientists will use 0.1, 0.7 micron as the cutoff point because they will use a GFF filter. The GFF is a glass fiber filter. Glass fiber filter, the grade is F, which gives you a 0.7 micron. Sometimes they use a millipore uh, nitrocellulose filter that will give a 0.45 micron. So this is familiar to you because you all use this to do your MFT. So this is just basically to divide your water samples into the particulates and the dissolved. So what are the particulates in the sea? That will be your microorganisms. You have your you'll have your chlorophyll, your so for biological organisms, the division is uh, usually you start with 0.2 micron, then you have your uh, then you have your 2 micron size, then you have your 20 micron, then anything above 200 micron. Here, whatever that's trapped on your 0.2 micron, that's your bacteria. That whatever that's trapped on the 2 micron filter, that's your protozoa. Whatever that's trapped on 20 micron and above, that's your micro zooplankton. And then whatever that's above, usually those are your macro plankton. They can be your copepods, your diatoms, etc. Then anything that's 0.02 micron captured will be your virus. So this is how biological oceanographer will separate the biological samples according to size. Okay? Remember, this is not absolute. Okay? Okay? Because some bacteria will still pass through this filter. Some bacteria will still be trapped in this filter. But we use this designation to make our life easier. If not, there is no way we can study the different components of the organisms in the sea. Okay, so this is how we separate them out. So if you want to know the term for this, so anything less than 0. Around this range, that's your femtoplankton. Then here will be the next one will be your pico picoplankton. Picoplankton will be your bacteria, your cyanobacteria. Your protozoas are usually your nanoplankton. Okay, then you have your microplankton and macro. Okay, so these are the terms that you can use to describe the different fractions. Any questions?